In this lecture, we're going to begin with an overview of the process in which skeletal muscle is recruited and activated to produce force. And we begin by examining the energetic requirements and events that take place inside a skeletal muscle, particularly three proteins that are relatively large consumers of energy inside the skeletal muscle cell. And when we talk about energy consumption, this all centers on the use of ATP as a source of potential energy. And to harness the stored chemical potential energy in an ATP molecule, it must be broken up in terms of releasing the inorganic phosphate molecule. And in doing so, the chemical energy that held the inorganic phosphate to ADP is transformed into heat and usable energy. That usable energy is mechanical in nature in the sense that molecules or other large proteins will be moved. And so in order to do that, in order to cause this ATP molecule to break up, an enzyme has to mediate this process so it can work very, very quickly. And that enzyme is an ATPase. And there are three major ATPases in the muscle cell that are large consumers of ATP. The first that was discussed in an earlier unit is the sodium potassium ATPase. And here you see a reconstruction graphic of the sodium potassium ATPase embedded in the membrane of the muscle cell. And the function of this ATPase is to bind three sodium ions inside the cell and to move those three sodium ions to the outside of the cell where the majority of sodium ions exist in a resting state. Once that process is completed, the protein will form a chemical bond with two potassium ions and move those two potassium ions towards the interior of the cell where they naturally exist in higher concentration under resting conditions. But in order to do that, the protein will also bind an ATP molecule, and that ATP molecule will be hydrolyzed and the stored chemical energy will be released, thereby transforming it to mechanical energy to move the ions back into a state of resting equilibrium. Another protein that will be discussed in a little bit in a later segment is what we call SERCA. And SERCA is a protein embedded not in the exterior cell membrane, but within the sarcoplasmic reticulum membrane, which has a similar structure to the cell membrane. So SERCA is an intracellular protein that traverses both the cytoplasm and the lumen of the sarcoplasmic reticulum. And so the function of this protein is to bind to calcium ions in the cytoplasm and to physically move those two calcium ions towards the interior of the sarcoplasmic reticulum. And as this particular protein is an ATPase, it will do so through the hydrolysis of one ATP molecule. So it will simultaneously bind an ATP in the cytoplasm, facilitate a chemical reaction that will harness and release and transform the stored chemical energy in ATP so as to transform it to heat and to transform it into a usable form of mechanical energy and move those two calcium ions back into storage in the sarcoplasmic reticulum. The third ATPase that is a large consumer of energy inside the skeletal muscle cell is the myosin ATPase. And the myosin ATPase plays an important role in force production. So the structure of this protein is such that the what are called globular heads, extensions of two long polypeptide chains wound up around each other, will form chemical bonds with another protein called actin. And in order to produce force, to go through the cycle of force production and mechanical relaxation, an ATP molecule is harnessed, hydrolyzed, and the stored chemical energy is uh, released, and that will allow the repeated production of force. And so these three proteins are typically active all the time in the skeletal muscle cell, but when an alpha motor neuron activates that skeletal muscle cell, the activities of these three proteins increase considerably. And on this slide, we'll review the process in which that skeletal muscle cell is activated by the distal component of an alpha motor neuron. So recall that the activation of an alpha motor neuron begins in the spinal cord, and that action potential uh, uh, passes along the entire length of the very long axon, and that's what we see illustrated at the top of this slide. So we see an action potential propagating along the full length of the axon until it reaches the terminal part of that axon, and it will trigger the conformational change, which is to say that voltage-gated calcium channels will change shape slightly, and when doing so, will allow calcium, which exists in higher concentrations in the extracellular medium, and because of the higher concentration, calcium will flow downhill and into the cytosol of the terminal axon. And that process will facilitate fusion of intracellular, what are called synaptic vesicles, with the interior of the membrane of the terminal axon. And exocytosis will ensue, in which that fusion of the vesicle to the membrane will see the stored neurotransmitter, acetylcholine, will be exocytosized, for lack of a better term, into the extracellular space. And these acetylcholine molecules will migrate through and find chemical binding sites on proteins on the postsynaptic membrane. And these are the ligand-gated sodium channels. And when the neurotransmitter connects to the ligand-gated sodium channel, the channel will open and allow the downstream very fast movement of sodium ions, which are held in higher concentrations in the extracellular medium, downhill towards the interior of the cell. The next step is that another protein embedded in the muscle cell membrane, acetylcholine esterase, ASE, indicating that this protein is an enzyme, will facilitate the binding of acetylcholine molecules and degrade those acetylcholine molecules into choline and acetic acid. Choline is then taken back up into the terminal axon and is reconstituted into new acetylcholine molecules, packaged up into synaptic vesicles and prepared for another wave of action potentials if that alpha motor neuron is activated. So to review these steps here with number 1, and this corresponds to the numbers in the diagram on the right-hand side, we see voltage-gated calcium channels are opened by a change in the electrical state across the membrane. And recall, the action potential will cause the interior of the membrane to, over a very short period of time, become positive. And that change in the electrical state is enough to cause the calcium channels to open. Once the calcium channels open, the intracellular concentration of calcium will increase, and that will drive vesicle fusion to the membrane through connections to those vesicle proteins triggered by the opening of those voltage-gated calcium channels. That will lead to numbers 3 and 4 on the diagram, in which fusion on the intracellular side of the synaptic vesicles will see acetylcholine released into the extracellular space, and that amount is referred to as quanta size. And one packet, one synaptic vesicle, can contain anywhere from 5,000 to 10,000 acetylcholine molecules, and one action potential will cause the release of anywhere from 100 to 300 vesicles, each of which contain anywhere from 5,000 to 10,000 acetylcholine molecules. And this is a very fast event that occurs over fractions of a millisecond. The very large release of these neurotransmitters functions to increase the probability of ligand binding to the postsynaptic membrane sodium channel. That increased probability with many of these acetylcholine molecules will tend to cause that postsynaptic membrane to reach what's called an end plate potential. And the end plate potential is exactly the same as an action potential, but it's specific to being caused by the neurotransmitter, and then that will lead to opening of voltage-gated sodium channels along the rest of the postsynaptic membrane, and the process ceases when the action potential of the motor neuron or when the membrane potential of the terminal axon returns back to its resting state, and acetylcholine esterase degrades acetylcholine. That will cause closure of the ligand-gated sodium channel, and choline and acetic acid are removed. And then choline, to finalize steps 8 and 9, choline is used by the presynaptic cell, in this case the terminal axon, to make more acetylcholine molecules. Now, how this ties together with force production is that we see, once again, we start with the axon terminal, and that axon terminal, once an action potential has propagated to this point, will cause that release of neurotransmitter through primary and secondary synaptic clefts. The secondary synaptic clefts refer to the space between the terminal axon and what are called junctional folds in that very isolated region of the muscle cell membrane, also referred to as the motor end plate. The entire connection of the terminal axon and that isolated region within the proximity of that terminal axon is just simply referred to as the neuromuscular junction. And acetylcholine, as a review here, is released from the terminal axon. This neurotransmitter will bind to nicotinic acetylcholine receptors, which is another term for the ligand-gated sodium channel. Those terms are used interchangeably. And that will generate the end plate potential, which is the term for the action potential that takes place within the vicinity of the alpha motor neuron as triggered by the neurotransmitter. And that leads to, as indicated by these arrows, moving away from the neuromuscular junction of the action potential. So the end plate potential is generated by neurotransmitter. The action potential is generated by a change in the voltage across the membrane. So the EPP comes first. And we see here that embedded not just within the junctional folds, but also within the primary synaptic clefts, which are the closest regions across the terminal axon, are the ligand-gated sodium channels, also referred to as the nicotinic acetylcholine receptor. And what we see here, that action potential will propagate as it did along the axon from the spinal cord all the way to the neuromuscular junction. But the skeletal muscle cell is so designed that it has invaginations. So the muscle cell membrane is pushed towards the interior of the cell into what are called transverse tubules. This is still extracellular medium, as you can see here, and the green just indicates that this is cell membrane structure that is continuous but moves towards the interior of the cell. So the action potential propagates along the membrane and will continue propagating along the membrane, where it will trigger the release of calcium ions housed inside the muscle cell. So contrasting with the movement of calcium ions into the terminal axon when triggered by the axon action potential, those calcium ions up here come from the extracellular medium. These calcium ions come from storage intracellular within the sarcoplasmic reticulum, which is a tubular network that houses many calcium ions. And when calcium is triggered to release because of the action potential of the muscle cell membrane that travels down the transverse tubule system, calcium will move from storage into the cytoplasm, and as such will interact with contractile proteins. Normally, a protein called tropomyosin, which will be discussed in more detail in a subsequent video, will block binding sites for the myosin ATPase. But calcium will bind to another protein called troponin C. By binding to troponin C, it will change the shape of it and move tropomyosin out of the way so that the myosin molecule can now bind to these actin binding sites, and each of these small circles, spheres, represents a single actin molecule. And that is the cause of force production. Now, in order to stop force production, two events have to take place. Action potentials have to stop along the alpha motor neuron. And calcium has to be removed. And when calcium is removed from its connection to troponin C, that tropomyosin will then move back and block the binding sites, preventing force production. To do that, calcium has to be actively transported into the sarcoplasmic reticulum. The reason it has to be actively transported is that calcium, even though it is released, is still in higher concentrations in the sarcoplasmic reticulum than it is in the cytoplasm. So calcium ions have to be moved uphill. So it takes more than just opening a channel to do that. And what does that is a protein embedded in the membrane of the sarcoplasmic reticulum, which is referred to as SERCA, that was introduced on slide number 2, that we'll discuss in greater detail in a subsequent video. So this ends this introductory portion of Unit 3, Muscle Physiology.